So we're going to be discussing renal physiology. So we're going to be focused on the structure and the function of the kidneys, more so the function, specifically looking at that structural or functional unit of the kidneys, the nephron. So I'm going to guide you through chapter 19 of our book, where we're going to explore how essentially the kidneys help to regulate fluid balance, blood pressure, and waste excretion. Okay? So the kidneys perform several critical functions in the body. These are going to include regulating of extracellular fluid volume and blood pressure, regulation of osmolarity, maintenance of ion balance, especially for like sodium, potassium, calcium. We also have the homeostatic regulation of pH, right? So we can remove or conserve hydrogen ions or bicarbonate ions. We're going to help, or the kidneys are going to help to excrete waste. And also don't forget about the hormone function of the kidneys, right? So the kidneys also produce hormones, right? Or its endocrine function, right? In producing hormones, number one, we're going to produce EPO, or erythropoietin. Erythropoietin is going to be an enzyme that will stimulate more red blood cells to be produced, right? So it regulates red blood cell synthesis. Another hormone is going to be renin. Renin is going to be part of that renin-angiotensin-aldosterone system. And essentially what renin does is it helps to increase blood pressure, right? So your kidneys will respond to a drop in blood pressure by secreting renin. And we're going to talk more about that in chapter 20. Another hormone that you don't see here, but I'll write it out, I'll put number 3, would be vitamin D, calcitriol. So the kidneys will produce that biologically active form of vitamin D, calcitriol. So when making vitamin D, and we'll talk about this, it requires a couple of different organs. It requires your skin, it requires your liver, and it also requires your kidneys, okay? So just going over a little bit of the anatomy of the urinary system. So it's going to consist of paired kidneys, right, that I just underlined. That's going to essentially filter the plasma to produce urine, right? In the kidneys, we're going to have these functional units called nephrons that's going to be doing the work of filtration, reabsorption, and secretion, okay? And then we have these tubes, right? These tubes, these are called the ureters, right? And the ureters are going to carry the urine from the kidneys to the bladder. Then in the bladder, we're going to store that urine, okay? From then, we're going to be able to excrete that urine through the urethra to go out of the body, right? So in females, the urethra is relatively shorter, and it's going to exit near the vagina. In males, it's going to be a little bit longer, and it's going to exit via the tip of the penis, okay? So looking at the structure of the kidney, notice how we have 2 layers. We have a medulla layer that I'm covering right there, and then we have that outer cortex layer, okay? So you got to understand that. An inner medulla and an outer cortex layer. Now, because the kidneys are going to be arranged in these 2 layers, the outer cortex is actually going to contain 80% of the nephrons. These are going to be called cortical nephrons. Cortical meaning cortex, all right? The inner medulla is going to contain 20% of the nephrons, and these are going to be called juxtamedullary nephrons, right? So they're going to be right near the medullary region. And we'll look at the different nephrons in just a second. Also, when we're looking at the structure of the kidneys, we got to look at the blood vessels too. These blood vessels will form a portal system. So we talked about other portal systems, and this is going to be the renal portal system, where we have a capillary system connected to another capillary system. So looking at the blood supply to the kidney, so blood is going to enter the kidney via the renal artery. Ultimately, it's going to go through a couple of different vessels. Then it's going to reach the afferent arteriole. From the afferent arteriole, blood is then going to enter the glomerulus capillary, then the efferent arteriole, and then the peritubular capillary. But notice how we have glomerulus capillary connected to another capillary, the peritubular capillaries, okay? That's what it means to be a portal system, okay? And a capillary bed that's connected to another capillary bed. So looking here at the structure of the nephron, and we'll look at this a little bit more, right? Again, 80% of these nephrons are going to be cortical nephrons, right? And then 20% is going to be juxtamedullary nephrons, okay? And so if we look here at, we can see the difference between these two nephrons, I'm sorry, it's nephrons. So you can see the difference between these two nephrons, right? I'm hoping I'm saying nephrons and not neurons, right? But looking at these nephrons, the nephrons again are going to be the functional unit of the kidney. So notice how this loop of Henle is shorter than this loop of Henle, right? So this juxtamedullary nephron has a much longer loop of Henle than the cortical, right, nephron, okay? And so I'm going to zoom in a little bit, right? So we can see here, as blood enters, blood is going to enter via the afferent arteriole. And so let me actually go over here, right? Blood enters this nephron via the afferent arteriole. Then that blood is going to enter the glomerulus capillaris, and this glomerulus capillaris is going to be within this renal corpuscle region. Blood will then go through here, right? And then it exits via the efferent arteriole. And notice how this vessel then branches off to form a capillary system, right, which is called the peritubular capillaries. And the peritubular capillaries are going to be surrounding the tubular system of the nephron, right? Notice how it's surrounding the tubular system of the nephron, whereas the glomerulus capillary is only located in one region, and is located in the renal corpuscle region, okay? So understand the difference in where these capillaries are going to be located. So the function of the renal portal system really, so we're trying to filter out the blood, right? We're filtering out the blood, and it's going to go into the lumen, right? So we're filtering out the blood, and it's going to go here. It's going to go into the lumen of the nephron, specifically that Bowman's capsule, okay? And then what we're going to do is, so that's job number one, right? Filtration. The next thing we're going to do is we're going to reabsorb that fluid right back into the tubule, or right back into the blood supply system from the tubule, okay? So even right here, so let me zoom in right here, and the proximal convoluted tubule, so that fluid went into the Bowman's capsule, now it's in the proximal convoluted tubule, and then one of the other functions is reabsorption, and so whatever gets filtered out, not everything, but certain things will get reabsorbed back into the blood at the peritubular capillaries. And if it's going back into the blood, it's going back into the body. So you have to understand whatever stays in this tubular system is going to exit the body, okay? But if we reabsorb it back into the blood, it stays in the body, okay? So you got to remember that. And when we're looking at, a nephron, one nephron will have two arterioles, afferent and efferent, and two sets of capillaries, the glomerulus capillary and the peritubular capillaries, right? There's going to be around 2 million nephrons per kidney, okay? So let's look at the structure and the parts of the actual nephron. So we know the glomerulus, that's going to be the capillary bed, right? The capillary system, right? Where we're going to be filtering out the blood. Okay, where we're going to be filtering out that blood is going to be into the Bowman capsule space. So this capsule space right here, right, that's surrounding the actual glomerulus, that's called the Bowman's capsule space, and that's where that fluid then gets filtered into. Together, the glomerulus and the Bowman's capsule is going to be referred to as the renal corpuscle. Okay, so that's where we're going to have filtration at, and that's renal corpuscle. Then that fluid is not called blood now, right? Whatever gets filtered through, we're going to call that filtrate, okay? That filtrate will then move through the tubule, right? Move through this nephron, okay? And so when we're looking at the different parts of the nephron, the part of the tube that's stemming right off of that Bowman's capsule is going to be called the proximal convoluted tubule. Proximal meaning it's close to, okay? So the proximal convoluted tubule, then we go into the loop of Henle, right? This loop of Henle will have a descending limb, and then ascending limb. Okay, from the ascending limb, then we have the distal convoluted tubule, and then the distal convoluted tubule is going to dump out into the collecting duct. Okay, so multiple nephrons will then dump out into the collecting duct. From the collecting duct, that filtrate, right, is then going to be called urine. So after that collecting duct, now it's called urine. That urine would then move into the renal pelvis, then go to the ureters, then go to the bladder, then go to the urethra, then out of the body. Okay. So here are the different functions of the kidneys, specifically when we're looking at the nephrons, right? So the kidneys will filter, reabsorb, and secrete. Filtration, like how we just looked at, is the movement of fluid from the blood into the lumen of the nephron, right? And so this filtration is going to occur at the renal corpuscle, right? We're filtering out the fluid from the glomerulus into the Bowman's capsule. And now that fluid that gets filtered out is now, again, called filtrate. Another function of the kidneys would be reabsorption. Reabsorption is the movement of material, or the movement of that filtrate, right, from the tubule back into the blood. Specifically, back into the blood at the peritubular capillaries. Okay, so filtration is going to occur within the renal corpuscle. Reabsorption is going to occur along the peritubular capillaries. Secretion is going to be a selective process from which materials are going to move from the blood into the lumen of the tubule. And this will also occur along the peritubular capillaries. Okay? So I kind of like this image a lot right here, right? Again, filtration only takes place in the renal corpuscle. This is filtration right here, right? Only in the renal corpuscle from the glomerulus into the Bowman's capsule. Notice how, filtration is going to happen freely. If it can fit through, if it can pass through that filtration membrane, it will get filtered through, right? And so reabsorption, going from the tubule back into the blood. Whereas secretion, that selective process, is going from the blood back into the lumen, right? Back into the lumen. So filtration and secretion go in the same direction, from the blood to the lumen, whereas reabsorption is going to go in the opposite direction because you're reabsorbing it back into the body, going from the tubule back into the blood. Okay? So when we're looking at that filtrate, it's going to be similar or almost identical to plasma at the renal corpuscle, right? Your kidneys or your nephrons are going to filter 180 liters per day. Okay, 180 liters per day. About 70% of that filtrate that you just filtered through is going to get reabsorbed right back into that proximal convoluted tubule. So you filtered it, right? But you're going to reabsorb most of it back, right? Almost immediately. Okay, so when we're trying to understand, well, how much fluid was actually excreted out of the body, right? We can use this equation, right? So the amount of solutes that are excreted will equal the amount that was filtered through minus the amount that was reabsorbed back into the blood plus the amount that was secreted, selectively secreted. Okay? So again, the amount of fluid or the amount of solutes that are excreted out of the body will equal the amount of fluid that was filtered minus the amount that was pulled back into the blood plus the amount that was selectively secreted out. Okay? So this just goes over the renal corpuscle. Now I kind of like this image right here, and we'll see this image again, right? But this is the glomerulus capillary right here, where that blood is going to be at, and then it's going to get filtered through. It's going to get pushed through these membranes, right? And it gets into this Bowman capsule space. Okay? And so when we look at what's covering, essentially, this glomerulus capillary, the capillary, and remember, capillaries, these are just like the smallest of the blood vessels, right? And so you're going to have these podocytes. And you can see these podocytes right here, right? And these foot processes that you can see right here, right? They're basically surrounding each of the capillaries, leaving slits through which like filtration can actually take place. And then you have this mesangial cell right here that are going to be between the capillaries. That's going to constrict, basically altering blood flow. Right, controlling like the diameter and the radius of the blood flow, okay? And that can promote and alter filtration rate. So for filtration to occur, we know that it's going to be the free mass movement of materials. where substances, they're going to pass through 3 barriers before entering the tubule lumen. So number 1, that fluid, right, has to go through the glomerular capillary endothelium, so the wall of the actual blood vessel. And remember, these are going to be fenestrated capillaries, so they're going to be porous, okay? Now, it has negatively charged pores, right? So these negatively charged pores are going to repel those negatively charged proteins and red blood cells, meaning red blood cells and proteins are not going to go through, right? They're not going to be able to go through because they're repelled. Number 1, that's the first barrier. The second barrier is going to be the basement membrane, right? When we're looking at this basement membrane right here, right? So that's also going to exclude most of those plasma proteins. So again, proteins are not going to get filtered through. Then you have the podocytes, right? You have the podocytes that I'm drawing in, right? These podocytes are going to have these processes, and it's going to allow for filtration slits. And this is going to be a part of the Bowman capsule epithelial wall, right? So these podocytes, they will create these slits, and if it's too large, it's not going to fit through. And of course, these mesangial cells, that's going to influence the filtration by altering the surface area, okay? So we have multiple barriers for this filtrate to actually go through, or multiple barriers for this fluid to actually go through to get filtered, right? Again, you're not going to find large things such as cells like red blood cells, white blood cells. You're not going to find large things such as plasma proteins like albumin. You're not going to find that. Globulins. You're not going to find these proteins, right, that are going to get filtered through. You will find like solutes, right? So sodium, chloride, calcium, potassium, things like that. Things that are smaller, solutes. You're going to find that gets filtered through because it can fit. So the filtration fraction is just simply the percentage of total plasma volume that filters into the tubule. So 100% of the fluid, 100% of the blood that enters via the afferent arteriole is going to enter the glomerulus. Only 20% of that enters the glomerulus will get filtered through. 80% of it that does not get filtered through will return back to the efferent arteriole, then go to the peritubular capillaries, then go to the renal vein, out, and then this process just gets, goes over and over and over. But of that 20% of fluid that was filtered, we're really going to reabsorb more than 19% of it back into the blood at the peritubular capillaries. So that leaves less than 1% of the volume that actually went through. Less than 1% of this 20% will actually get excreted out of the body. Okay? So there are some pressures that will influence filtration. So there's 3 main pressures that will influence glomerular filtration. We have the capillary blood pressure. This is going to be the hydrostatic pressure. This is the blood pressure within the actual glomerulus. And so this is going to favor filtration. So this is essentially blood pressure, right? And this favors filtration. So when we're looking at hydrostatic pressure is a pushing force, right? So it's going to push fluid out of that area, okay? And if it's pushing fluid out of that area, it's favoring filtration. Then we have the capillary colloid osmotic pressure. And remember, osmotic pressure is a pulling force. So this pulls fluid back into that area. And if it's pulling fluid back into that area, it opposes filtration, right? So it's pulling fluid back into the plasma. Thus, it's going in the opposite direction of it going into the Bowman's capsule, right? And so this capillary colloid osmotic pressure is going to be due to proteins, right? Such as like albumin, okay? And then another pressure that we have to consider is the capsule fluid pressure. And this is the hydrostatic pressure inside the Bowman's capsule. And again, it's hydrostatic pressure. So it's pushing fluid out of that area. So it's pushing fluid out of the Bowman's capsule into the blood vessel, the capillary, right? The glomerulus capillary. So that opposes filtration, right? So when we look at it right here, you have one pressure that promotes or favors filtration. That's the hydrostatic pressure or the blood pressure of the Bowman's capsule. And then you have these other 2 pressures that opposes filtration. So you have the capillary colloid osmotic pressure that's pulling fluid back into the blood vessel, right? And then we have the hydrostatic pressure of the Bowman's capsule that's pushing pressure away or pushing fluid away. So these two pressures are going or opposing filtration, whereas the hydrostatic pressure of the actual blood vessel favors filtration. And again, filtration is movement of fluid out of the blood vessel into the Bowman's capsule. Okay. So when we're looking at these pressures, right, we can calculate your glomerular filtration rate, right, or your glomerular filtration pressure. Speaking of glomerular filtration rate, simply it's a rate. It's simply the volume of fluid that is filtered per unit of time. So it's the amount of fluid that is filtered in a minute, basically. That's going to be influenced by the net filtration pressures that we just discussed and the filtration coefficient, meaning the surface area of the glomerular capillaries available for filtration and the permeability of those filtration slits. Now, glomerular filtration rate is going to be relatively constant, right? Your GFR, your glomerular filtration rate, is going to be controlled primarily by regulating blood flow through the renal arterioles. So increased resistance in the afferent arteriole will decrease glomerular filtration rate, whereas increased resistance in the efferent arteriole will increase glomerular filtration rate. And we'll look at this in a second as well. Now, when we're looking at your blood pressure, so blood pressure can range, right? Your body has the ability to auto-regulate itself, right? To maintain a constant glomerular filtration rate. So this auto-regulation maintains a constant glomerular filtration rate, again, of 180 liters per day. So we're maintaining a constant glomerular filtration rate over a wide range of blood pressures, as we can see right here, right? And these blood pressures, essentially hydrostatic pressure, is going to drive glomerular filtration rate. So even if your mean arterial blood pressure is, 80, 120, 160 or so, right? That blood pressure is high, which means your glomerular filtration rate would be high. Your body is able to auto-regulate itself and maintain a constant glomerular filtration rate of 180 liters per day, okay? Okay. So here, let's look at how your body essentially will auto-regulate itself, right? So as we see here, as resistance changes in like the renal arterioles, it can alter renal blood flow and your GFR. So if we look at the glomerular filtration rate right here, right, that's simply the amount of fluid that's being filtered per unit of time, okay, from the glomerulus into the Bowman's capsule. So vasoconstriction, essentially of the afferent arteriole, it increases resistance right here. And so if you're putting more resistance on that vessel, not that much fluid will enter the glomerulus, right? If we're holding this basically, so if you're putting your hand over here and you're constricting it, right, that decreases the amount of fluid that goes into the actual capillary, into the glomerulus capillary. So if not that much fluid is in there, you have a reduced hydrostatic pressure, which will mean you will decrease your glomerular filtration rate, okay? On the other hand, if you constrict the efferent arteriole, right, that means less fluid actually leaves the glomerulus, more fluid kind of builds up in here, and if more fluid is building up in here, that increases the hydrostatic pressure there. And so, if you have increased hydrostatic pressure, that's going to increase your glomerular filtration rate. Okay? So I'm going to finish the rest of this in another video where I'm going to finish the rest of the chapter, okay? And I'm going to start off with the glomerular filtration rate being subject to autoregulation.